Welcome back. Well, in the previous lecture, we met Stéphanie Robert, who talked about plants' ability to adapt their physical form to better cope with rapidly changing environments. Now we're going to take a close look at the soil. So please welcome Professor Paul Kardol.
Growing up in a small town surrounded by forests and heathlands, I've always been attracted to nature and the complexity of nature. And that sort of triggered my interest in trying to figure out how nature works. My research broadly focuses on how global change influences the interactions between plants and their associated soil organisms, and what it means for the ecosystem processes they drive. My name is Paul Kardol and I'm a professor in forest microbiology. I work with soil microbes, but also with soil animals, like nematodes. We conduct mechanistic experiments using climate chambers and greenhouses, where we can manipulate temperature and water availability. But we also explore larger scale patterns using environmental gradients, for example, forest alpine ecotones. I'm motivated by learning new things every day, by finding answers to questions that nobody has asked before, and by working at the forefront of a globally important field of research. I'm also motivated by the interactions with the diverse community of people in this field. I hope to achieve a better understanding of how ecosystems will respond to climatic changes, in particular the underlying mechanisms, the species interactions. But I also hope that my research informs on how to adapt forest management to climate change and make our actions more sustainable.
Welcome, Paul.
Thank you.
What excites you about studying soil ecology?
Oh, soil ecology. There's so much to get excited about. One of the things certainly is the very high diversity of soil communities. There's so many species doing all kind of different things. Like the below ground is almost like a world on its own, with positive and negative interactions amongst different organisms from a wide range of different taxonomic groups. But then at the same time, of course, also the below ground world is very strongly connected to the above ground world. So everything what you see above ground is somehow, in one way or another, connected below ground. So it's the soil communities, for example, they are needed to continuously cycle nutrients and energy through the system that makes plants grow. But then at the same time, there's also like soil pathogens and root herbivores that have negative impacts on plant growth. So there's so much going on below ground, and we're discovering more and more and more. And at the same time, there's still like so many unanswered questions. So I think that's quite exciting.
Yeah. And you mentioned in the video that you're driven by discovering new things every day, by finding answers to questions nobody has asked before. I think that was exactly what you said. And I wonder, what personal qualities do you think are important to be able to break new ground in research, to ask fresh questions, and to look at things differently in such a complex research that you are doing?
That's a good question. Of course, you need to be curious and really have the drive to want to know how things work. You need to sort of be often I think a little bit differently or out of the box. That can be thinking big, but also sometimes thinking very small. So yeah, it's a bit like systems thinking. How do species and communities interact, and then how do they function in the context of a larger system? I think it's also good to be able to conceptualize things and synthesize things, to always see the bigger picture, the wider context of your research. Yeah, and then maybe a bit more practical, the kind of research that we do, which is often in the field, and things do not always go as planned. So being pragmatic or adaptable is yeah, I think that's very important. You always have sort of a plan B to do things a different way or analyze data in a new way. So just, yeah.
Ah, thank you. And how do you think you can do to foster a culture like that in a group of research where this can develop or?
Several things. First, it helps a little bit what others have been talking about. If the group consists of a diversity of people coming from different backgrounds with different experiences, that's it's personal backgrounds, but also research backgrounds. Having studied in different ecosystems, you come up with different ideas, different ways to make connections or analyze things in a different way. But also, of course, important to have a culture in a group, lab group, or a large group of people collaborating in a research project to have an environment where people feel comfortable to express their opinions and be open for discussion and feedback. Yeah, and then maybe also when you design sort of new research, when you write grant proposals, there's always try to leave a little bit of room for the people who are then actually often start working on the project. It can be PhD students or postdocs, to have a little bit of freedom, academic freedom that we talked about before, to make your own sort of choices, to apply your own ideas to the broader research questions that we are working on.
Yeah, well, thank you. Why don't you enter the stage and prepare for a lecture?
I will do.
Yeah, thank you. And I have the pleasure of introducing Paul Kardol, Professor of Forest Microbiology, giving his lecture under the title Breaking New Ground in Climate Change Ecology, What Happens Below the Ground Matters.
Thank you. Our planet is warming up. It really is. Average temperatures have risen a lot since the Industrial Revolution, particularly in the last couple of decades, and projections indicate further increases in the coming years. This graph shows the temperature anomalies with reference to the pre-industrial temperatures. That's so how much warmer it has gotten since. And if you would just focus or look at the red line and the yellow line, you see that we are now basically already above the 1.5 degrees Celsius threshold to which countries aim to limit global warming under the Paris Climate Agreement. Then why is our planet warming up? It's largely because of the increased levels of carbon dioxide, CO2, and other greenhouse gases in our atmosphere, largely resulting from human activities. So knowing that, I'd like to take you on a little tour to one part of the research that we are doing in our group, focusing on terrestrial ecosystems under global warming. Terrestrial ecosystems can mitigate global change by taking up CO2 from the atmosphere. But the question is whether or not this will actually happen. On the one hand, plants fix CO2 from the atmosphere, but ecosystems also release a lot of CO2 through respiration by the plants, and also by the soil organisms. So when it comes to carbon cycling in an ecosystem, it's a delicate balance between carbon uptake and carbon release. So when we talk about ecosystem carbon exchange, we often ask the question, is it a carbon sink or is it a carbon source? In this talk, I would first like you to walk you through a couple of points that are sort of key to the research that we are doing. First, to understand how ecosystems function, we need to understand or consider the interactions between what happens above ground and what happens below ground. The things happening above ground, like plant growth, photosynthesis, depend on the things happening below ground, like microbial activity, soil pathogens, and vice versa. The things happening below ground happen on the things above ground. For example, like dead plant leaves, they decompose in the soil. Second then, when it comes to global warming, effects are very different above ground compared to below ground. And I will show an example of that later. Third then, in studying the effects of warming, we do not just compare warm versus cold or current versus future, but we explicitly look into non-linear temperature responses. And the little figure on the right illustrates why. In biology or ecology, plants and soil organisms typically do not respond linearly to a change in temperature. Species have temperature optima and lower and upper threshold or tolerance levels. And if you would then look at the functioning of a whole ecosystem, this means that we may detect certain temperature thresholds or tipping points where all of a sudden things change. Maybe a few steps back, I got interested in plant-soil interactions during my PhD at the Netherlands Institute of Ecology. This is a picture of one of my very first greenhouse experiments that I did during that time, looking at feedbacks between plants and soil microbial communities. And I think that further triggered my interest in life below ground. It's really fascinating. It's full of life. Down there, it's so many microorganisms, bacteria, fungi, but then there's also a lot of different little soil animals, each with their own role and function in the ecosystem. For example, my sort of pet organisms, nematodes, that I've been studying over the years. These are little transparent worms. Some of them feed on bacteria or fungi, others feed on plant roots, the root herbivores, and others feed on each other. And then all together, the microorganisms and the soil animals form a complex soil food web, which plays a key role in ecosystem carbon cycling. Then back to global warming. Here it is important to consider the differences between above ground temperature and below ground temperature. This here is an example of one of the research sites that we have in Abisko in northern Sweden. Here, as in many other places, we are measuring temperature below ground in the soil, at the soil surface level, a little bit above the soil surface level in the understory vegetation, and higher up at a tree trunk. This graph here shows 2 years of temperature data, and it illustrates two main things. The very strong seasonal fluctuations, and maybe even more important, how these fluctuations vary depending on where you measure the temperature. If we look at the soil temperature, that's the blue line here, you see that it actually doesn't really fluctuate very much. In the winter, here and there, it's almost always just around 0 under the snowpack. And in summer, it doesn't really go much higher than, what is it, like 10 degrees. But then if you look at above ground temperature, it goes down to -30 almost in the winter and +30 in the summer. So then the above ground plant parts experience completely different temperatures than the below ground plant parts and these soil organisms. So that's something really important to keep in mind if you want to understand how global warming would affect the functioning of ecosystems. So then what happens if we heat up our ecosystems? Here there's one sort of overarching hypothesis that we have been working with, posing that warming disrupts or decouples interactions among coexisting species of plants and soil organisms. Plants, they live together with good soil organisms, mutualists, and bad soil organisms, pathogens. And then when it's getting warmer, these relationships, they may strengthen or weaken, maybe they completely disappear, or be replaced by completely new relationships. And all of that would have consequences for the carbon cycling processes that they drive. How do we measure carbon fluxes? There's many different approaches, and Matthias already talked about it this morning. We are often using a very or relatively simple chamber approach. We seal the chamber to the soil surface, and then there's a probe that measures the change in CO2 concentration within the chamber. And from that, then we can calculate how much CO2 enters the ecosystem and how much CO2 leaves the ecosystem. Is it the sink or is it the source? Approaches like this we have used to answer a variety of different kind of questions in different ecosystems. This here is an example where we tested the consequences of plant diversity loss for ecosystem carbon cycling using a system of lake islands up north in Sweden. But then for our questions on how global warming affects ecosystem carbon cycling and how all of this is driven by the plant-soil organism interactions, we use forest alpine ecotones. These are basically mountain slopes, elevational gradients with colder temperatures at the top of the mountain, and then it gets warmer the further you go down. So this means when we go from colder temperatures, from alpine or subarctic tundra vegetation at the highest sites to forest sites down in the valley. And as such, we can use these forest alpine ecotones as a proxy for global warming. This is what one of our sites looks like above the tree line in northern Sweden, in Abisko, where we have been doing most of our research over the last 10 years. And this is what it looks like below the tree line. But we're not just working in Sweden. To get a sort of a broader understanding of how warming affects plants, soil organisms, and ecosystem carbon cycling, we apply the same methods, same protocols across a variety of different places around the world with similar temperature gradients, but with different plant species and with different soil organisms. And that allows to get to search for general patterns. At the same time, we learn about the importance of the local species and communities. So this is what the tree line looks like in Australia. Very different from Sweden. And this is what the sites below the tree line look like in Argentina. This is what the alpine zone looks like in Argentina. Very different from Sweden or from Australia. And this is Colorado. So we can test and measure a lot of things in the field, but for certain questions, we go from the field to the lab. There we can do more detailed, more mechanistic experiments in the greenhouse or using climate chambers. For example, in the lab, we can put little intact ecosystems collected from the field in chambers that we can program according to a wide range of different future temperature scenarios. As such, then testing for non-linear temperature responses. Or we can build our own ecosystems by growing plant species in the absence or presence of certain groups of soil biota, or in presence of soil communities representative for higher temperatures or for lower temperatures. And that allows us to directly test the consequences of potential asynchronous responses of plants and soil organisms to warming. This is an example of a little intact mini ecosystem, then later to be used in a climate chamber experiment by PhD student Kusuke Tanigawa. This is an example from a different kind of experiment, growing little mountain birch seedlings, testing them across a wide range of temperatures in the absence or presence of soil biota. That's an experiment done by PhD student Berit Oberholzer. So in conclusion, under scenarios of global warming, will ecosystems function as a carbon sink or a source? Big question, but there is no simple answer to this question. And biology or ecology certainly is not about simple answers. It really isn't. But we're making progress. We really do, step by step, sometimes big steps, often small little steps in the field and in the lab. And I think by every step we take, we learn more about how the complex interactions between plants and soil organisms may hold the answer to this big question. Then to finish, I would like to emphasize that in science and academic research is not only about what we study or what we do, but certainly also very much about how we do things, how things are done, and by who. And here, not only do many hands make light work, but also more diverse group of people are more creative, more innovative, and just more fun to work with. And with that, I'd like to thank you and all my colleagues and advisors and supervisors and students and all the technicians who have always helped me so well during all these years. Thank you.
Welcome to take the seat again, and thank you very much. Thank you. In your lecture, you I just want to remind you to ask questions. Also, those of you who follow the broadcast. In your lecture, you mentioned that you grow plants with or without different soil organisms. How do you do that? Could you tell us something more?
Yeah, we do. There's different ways to do it. One approach that we commonly use is basically go to the field, collect soil, then split it, 2 batches. We sterilize 1 batch of soil, and then grow plants on these sterilized soils, re-inoculate it with little or small amount of living soil containing the soil organisms that we are interested in. And then these could be collected from different places. What you can also do is grow plants on sterilized soils, then use another batch of soil to extract different groups of soil organisms and then add them back. And then you can actually grow them in the plants with soil organisms in all kind of different combinations. So you can design experiments in a full factorial design with different treatments, soils, soil biota, from cold temperatures, from warm temperatures, and different combinations. So that's one way of doing it. Another way, which is more difficult and requires more resources and time, would be culturing individual soil organisms or individual microbes, and then add these as treatments later in your experiment. But that's more complicated.
Well, it sounds quite complex, all the whole thing. But thank you. We have a question from Pernilla.
Thank you, Paul. So what happens when you make a clear cut and take away all the vegetation, or when it's really, really dry conditions or really, really wet conditions in the soil? What about the greenhouse gas emissions? What happens?
Typically, you would lose a lot of carbon from the soil. I think Matthias showed an example from the very young forest where there's more greenhouse gases or leave the system compared to how much is taken up, because you first need plants photosynthesizing to fix carbon again. When it comes to the climatic factors that you mentioned, it depends a little bit, of course, for microbes, clear cuts, for example, they can be really warm in the sun. They may like that, but at the same time, it might dry out. They would not like that. So microbial activity actually might be going up or down depending on the combination of temperature and moisture.
What do we need to understand about carbon exchange in ecosystems to predict if they absorb or release carbon, and how can we gain that understanding? It's two big questions, but if you can say something more about that.
It relates to the question from Pernilla. Of course, first we need to understand who is doing what when it comes to the species of plants and the species of soil organisms. And then second, we need to understand their relationships with the abiotic environment. In this talk, I spoke mostly about temperature, but there's of course also other environmental factors like moisture availability, nutrient availability, which may be equally or sometimes maybe even more important than temperature. And then third, and that's maybe the most difficult part, we need to understand how these relatively small scale interactions would scale up in time. Seasons, we have been talking about years, decades, successional stages, and also in space, like from when you go from pots and plots to forest stands or entire watersheds or landscapes. And then I think then we really need to combine our experimental approaches with detailed measurements on the spot and much larger scale measurements. For example, using flux towers.
Yeah, exactly. We have a question from the fourth row, I think. I think that this will be the last question for this.
Yes, you have shown so nice pictures of tree lines, and I'm wondering what climate change does to the position of the tree line, and if in case the tree line moves up, what does that mean for CO2 storage in alpine regions?
Good question. Typically, if it would be getting warmer, the tree line would move up, because trees are to some extent limited by temperature, how far up they grow on a mountain slope. That's relatively easy to predict. Much more difficult it would be to predict, and that's part of our questions, what would happen with the carbon. Of course, when you get like taller woody species in your system, there simply is more carbon above ground, but it does not necessarily mean that there's also more carbon below ground. And there's some nice studies done by others at SLU as well, when actually showing that the carbon below ground might be actually less. depending on the changes in the soil microbial communities, for example.
Okay, so with that, I would like to say thank you to you, Paul Kardol. A warm
Thank you.
applause. And for those of us that are in this studio, there will be a coffee break, and hopefully for those of you that are watching the broadcast as well. So we will have coffee, and we will be able to be back here at 3:10. So welcome back then. And then we will meet Markus Schmidt. See you then.